The Alzheimer's Revolution, New Treatments. The Alzheimer's Revolution, New Treatments, New Targets, New Hope. Today I would like to show the current status and future perspective of Alzheimer's disease research. I would like to thank all of you for watching this presentation. Here is my COI disclosure. Here are the abbreviations used in today's lecture. You do not have to remember them all, but it would help understand the disease mechanisms. AD stands for Alzheimer's disease, FAD for familial AD, SAD for sporadic AD, FTDP for frontotemporal dementia with Parkinsonism, A-beta for amyloid beta peptide, PE for pyroglutamate, APP for a beta precursor protein, MAPT for microtubule-associated protein, NEP for neprilysin, the major A-beta degrading enzyme in the brain, ISF for interstitial fluid, CSF for cerebrospinal fluid, GS for glymphatic system, SST for somatostatin or somatostatin receptor, GPCR for G-protein-coupled receptor. Here are pathological hallmarks of AD. These are senile plaques composed of aggregated A-beta that accumulate outside cells, and these are neurofibrillary tangles composed of tau protein aggregates that accumulate inside cells. This slide illustrates a brief history of AD research initiated by Alois Alzheimer in 1906, which was followed by clinical and pathological studies, biochemical pathology, and the Big Bang that identified FAD-causing mutations and FTDP-causing mutations in the 1990s. And we are in the middle of the next stage. A large number of FAD mutations have been identified, and essentially all of them result in pathological A-beta deposition, leading to establishment of cause and effect relationship. While FAD-causing mutations lead to A-beta pathology, tau pathology, and neurodegeneration. MAPT mutations cause tau pathology without A-beta pathology, indicating that both A-beta and tau play essential roles in AD pathogenesis, and that blocking A-beta pathology will halt subsequent changes in AD. We study FAD based on the presumption that the anti-FAD therapy should also prevent SAD. At present, 2 disease-modifying therapies have been approved. One is lecanemab, the basis for which was provided by the group led by Lars Lannfelt. The other is donanemab, for which my collaborators and I made an initial contribution. These therapeutic antibodies may exert additive effects when used together as their epitopes are distinct. Here is a target epitope of donanemab that is generated by conversion of A-beta 142 to A-beta 3, PE42. PE stands for pyroglutamate. This conversion results in the loss of 1 positive charge and 2 negative charges at the amino terminus of A-beta. Let me now briefly discuss limited proteolysis. It is cleavage of a peptide/protein at a specific peptide bond. When a given protease induces hydrolysis like this, you will get these 2 fragments. Then you will synthesize a short pentamer or hexamer peptide corresponding to the carboxy or amino terminus, conjugate it with KLH via cysteine, and immunize animals to generate antipeptidic antibodies specific to the proteolytic product. Here is an example, that is calcium-dependent autolysis of calpain-1. Peptide, A, is used to generate an antibody to the pre-autolysis form, and peptide, B, to the post-autolysis form. Here is a Western blot data showing that these antibodies selectively recognize the corresponding calpain forms. Now I would like to show that antibodies to a substrate are more sensitive than those to a protease because the number of the former is generally larger than that of the latter. We thus made an antibody that recognizes calpain-cleaved spectrin. Use of this antibody allowed us to resolve spatiotemporal calpain activation after transient ischemia like this at time 0, 15 minutes to 4 hours, 24 hours, and 7 days, accounting for the delayed neuronal death. We then entered the field of AD research by generating antibodies that recognize distinct amino and carboxy structures of A-beta. Here is a Western blot data of formic acid extracts from the aged human brain. You can see that a beta X42 with various amino terminal modifications is more abundant than a beta 140 and 42. This shows that a beta 3E and a beta 4F are minor species, indicating selective accumulation of a beta 3PE. This is Western blot analysis of a beta extracted from AD brains. There are two indications. First, a beta 3PE, 42 is the most dominant species, and the electrophoretic patterns of a beta 3PE and a beta 42 exhibit a monomer, dimer, and trimer or tetramer in a similar manner. Second, it is technically possible to generate an antibody specific to a beta 3PE. These findings provided D'Amatos and colleagues with the conceptual and technical basis for generating donanemab. Why did previous studies fail to identify a beta 3PE42 in the brain? Pyroglutamyl, PE, peptides are resistant to Edman degradation used to chemically sequence peptides. The reversed phase HPLC column needs to be heated at 50 degrees Celsius to elute A beta 142, whereas room temperature is sufficient to elute a beta 140 and 3PE40. So, it was necessary to use very stringent conditions for elution of a beta 3PE42, that is basic solvent, TEA, plus betaine, a low molecular weight detergent with the column heated at 50 degrees Celsius. We speculated that presence of a beta 3PE may be associated with a beta metabolism. We thus analyzed a beta 1-42 metabolism by injecting internally radio-labeled a beta into rat hippocampus and have identified neprilysin as a major in vivo beta degrading enzyme. We then tested our hypothesis that a beta 3PE42 may be metabolically more stable than a beta 142 in vivo. Whereas the half-life of a beta 142 was about 20 minutes, that of a beta 3PE42 was about 90 minutes. Therefore, a beta 3PE42 is much more resistant to in vivo degradation than a beta 142. This, at least in part, accounts for the selective accumulation of a beta 3PE42 in the AD brain. Previously, we and others have shown that there are three pools of a beta in the normal brain: ISF extracellular A-beta, E-A-beta, TS-soluble A-beta partially bound to ApoE and others, and guanidine hydrochloride soluble or insoluble A-beta. Here are the references. Accordingly, the insoluble A-beta is much more abundant than the soluble A-beta, which is more abundant than ISF A-beta. I would like to indicate that the insoluble A-beta is difficult to remove by transport-dependent clearance because they are not quite diffusible. Consistently, the insoluble A-beta is much more abundant than the soluble A-beta in the wild-type mouse brain. And neprilysin deficiency affects the quantity of insoluble A-beta in a manner greater than that of soluble A-beta. Importantly, neprilysin deficiency exerts only a little or no effect on the levels of neuropeptides in vivo. This is a graphical summary. As A-beta is produced by the sequential cleavages of intramembranous region of APP, it is reasonable that a large portion of A-beta is associated with the plasma membrane, especially in the lipid raft microdomain, and oligomerizes/clusters at the ganglia side as the scaffold. Thus, the majority of A-beta is retained in the insoluble compartment and degraded by membrane-bound NEP. In classical biochemistry, neprilysin was generally regarded as a relatively nonspecific peptidase capable of degrading a broad range of peptides. However, accumulating evidence now suggests that, in vivo, neprilysin preferentially degrades insoluble A-beta species. This apparent selectivity may reflect the fact that neprilysin is membrane-bound under physiological conditions, whereas classical biochemical studies largely relied on solubilized forms of the enzyme. It may also be attributable to its cellular localization. Neprilysin is enriched at the presynaptic terminals of excitatory neurons, whereas many neuropeptides are primarily secreted from inhibitory neurons. Let me illustrate schematically the metabolism of A-beta that involves transport and catabolism in a simplified manner. K1 is a rate constant of A-beta anabolism. K2 is a rate constant of A-beta transport via the glymphatic system. K3 is a rate constant of A-beta catabolism. and a beta aggregation results in a beta deposition. Here is a mathematical formulation of a beta metabolism. The equation becomes like this under the steady-state condition. If K3 is much larger than K2, the amount of a beta is determined by K1 divided by K3 based on this data. Besides, K2 is brain size dependent, whereas K3 is brain size independent. Therefore, catabolism rather than transport appears to play a major role in a beta clearance. However, I would like to emphasize that I am not simply a catabolist, and there are situations where K2 becomes greater than K3. At leptomeninges, where vascular A-beta was initially isolated and identified, K2 may be larger than K3. Thus, the glymphatic transport is not necessarily irrelevant in A-beta clearance in this case. Also, the age-dependent leakiness of BBB-K4 may need to be considered. In 2001, we predicted possible existence of AD-associated risk mutations in the MME gene, which encodes neprilysin. This prediction turned out to be true approximately 20 years later by genome-wide association studies that identified an intronic mutation before the initiation codon and another exonic mutation that causes M8V amino acid substitution. The M8V mutation induces phosphorylation of S6 by MEK, leading to reduced expression on cell surface and extracellular vesicles, and this results in reduced extracellular A-beta degradation. Consistently, neprilysin expression declines with aging in the murine hippocampus and human temporal cortex. We then examined the effect of neprilysin deficiency on A-beta deposition in the APP knock-in mice and found that this increases the relative amount of A-beta 3PE. We also analyzed the effect of neprilysin deficiency in APPTG mice using LCMS. Neprilysin deficiency led to generation of A-beta 3E and A-beta 3PE. We then found that neprilysin deficiency upregulates DPP, APN, and glutaminyl cyclase-like, all of which contribute to a beta 3PE formation. And this form of a beta is a target of donanemab. These observations indicate the neprilysin-donanemab connection. This represents one of the first cases in which experimental biochemistry successfully predicted human genetic findings relevant to AD, apart from the landmark amino acid sequencing of A-beta by Glenner and Wong, which ultimately led to the cloning of APPC DNA and the identification of familial AD causing mutations. You may think that apolipoprotein E may be another case because it binds to A-beta and facilitates its aggregation, but there are so many substances that bind to A-beta and facilitate its aggregation, so apolipoprotein E happens to be one of them. Let me describe our next-generation mouse models, APPNLF knock-in mice, in which the murine A-beta sequence was humanized, and Swedish and Bayreuther Iberian mutations were inserted. Another line, NLGF, contains the Arctic mutation that facilitates A-beta oligomerization. The NLF line starts to accumulate A-beta at about 9 months, mainly cortex and hippocampus. This is a three-dimensional imaging of A-beta pathology in the APP-NLF knock-in mouse brain. The green indicates A-beta plaques, and the red indicates blood vessels. This experiment was performed in the laboratory of Atsushi Miyawaki in RIKEN CBS. Atsushi is one of the first scientists that made FRET fluorescence resonance energy transfer applicable to detecting protein-to-protein interactions in living cells and tissues. The NLGF line carrying an additional Arctic mutation exhibits a beta pathology 3 times faster than the NLF line. This also shows that the Arctic A-beta is more pro-inflammatory. The A-beta amyloidosis is shown by blue, astrogliosis by green, and microgliosis by red. Here are the advantages of the knock-in mouse models. The APP knock-in mice have overcome the following drawbacks of mutant APP or APPPS1TG mice. Lack of relevant negative controls, mice that overexpress wild-type transgene aberrancies. Inapplicability of genome editing technology. Destruction of endogenous gene loci. Use of artificial promoters. Artificial endoplasmic reticulum stress. Artificial calpain activation. artificial neurodegeneration without tau pathology, other overexpression artifacts, for instance, APPSG-ABAR interaction. If you perform whole genome sequencing of AD models available from the Jackson Laboratory, you probably will publish a paper that will be cited forever. Use of the AP knock-in mice has shown that neprilysin deficiency selectively elevates insoluble A-beta. Another A-beta-degrading enzyme is IDE, insulin-degrading enzyme discovered by Dennis Selko's group. IDE selectively degrades soluble A-beta, and its deficiency exerts a small effect on A-beta pathology in the APP knock-in mice. However, a combination of IDE and NEP appears to affect A-beta pathology in a synergistic manner, indicating the possible presence of epistasis, which means an interaction between different genes. We then reported that a neuropeptide, somatostatin, regulates brain A-beta 42 through modulation of proteolytic degradation in 2005, but it was not easy to identify the target receptor because the somatostatin receptors are composed of a homodimer or heterodimer, and because there exist 5 receptor subtypes from SST1 to SST5, making the total number of possible combinations up to 15. We thus analyzed the effect of homodimer-heterodimer deficiency on the neprilysin expression using corresponding knockout mice. This effort has identified the SST1-SST4 heterodimer as a pharmacological target, although it took more than 10 years of work. You may still wonder why there are 15 combinations. As you see, there are 5 homodimers and 10 heterodimers, making the total number 15. Good news is SST1 and SST4 are co-expressed only in the cortex and hippocampus, indicating that a specific agonist or allosteric modulator would exert a relatively small systemic side effect. This slide illustrates that deficiency of SST1 and SST4 promotes a beta deposition via reduced neprilysin expression, consistently indicating that SST1-SST4 heterodimer is a candidate target for treatment of preclinical AD. Importantly, neprilysin deficiency exerts only a little or no effect on various neuropeptide levels in the brain, as shown in the previous slide. Here is a summary of these observations. Aging reduces neprilysin levels through somatostatin depletion, leading to the accumulation of insoluble A-beta, which subsequently triggers tau pathology and inflammation, ultimately resulting in neurodegeneration and dementia. Neuronal dysfunction and neurodegeneration would further exacerbate somatostatin depletion, establishing a positive feedback loop that may accelerate disease progression. Activation of the SST1-SST4 somatostatin receptor heterodimer may decelerate this pathogenic cascade by upregulating neprilysin expression. This table summarizes the advantages of targeting somatostatin receptors, which are GPCRs, compared with therapeutic antibodies. GPCR targeting medications are generally less expensive, lower in molecular weight, easier to deliver to the brain, pharmacologically more specific, and associated with fewer systemic side effects. My goal now is to find a place where I can create opportunities for developing disease-modifying therapies that halt the progression of AD during its preclinical stage, ultimately enabling a future in which AD can be prevented before the onset of cognitive impairment. Here I would like to change my topic to the A-beta-tau axis. Because tau pathology plays an essential role in the etiology of AD, FTDP17, and other neurodegenerative diseases, elimination of tau pathology is expected to decelerate the downstream deleterious events. Targeting tau appears to exert a broader effect than targeting A-beta, but it would not be sufficient to control subjective cognitive decline and spatial memory impairment that may be caused by A-beta pathology alone. We thus generated and analyzed mutant humanized MAPT knock-in mice. Here is the major difference between the APP and MAPT knock-in mice. In APPKI mice, only the A-beta sequence was humanized, and the pathogenic mutations were introduced to the loci. In MAPTKI, the entire MAPT gene was humanized, and the pathogenic mutations were introduced because mRNA splicing affects the physiology and pathology of tau protein, and because pathogenic mutations exist both in the introns and exons. By the knock-in strategy, we intend to avoid the overexpression artifacts. This shows the pathogenic mutations identified in the human MAPT gene. We first humanized the murine MAPT gene and then knocked in some of these mutations in the first trials and additional mutations in the second trials. These are the major lines of mutant MAPTKI mice. Those marked by yellow have been mainly analyzed. We introduced the S320F mutation because of its synergistic nature. I would like to emphasize that Naoto Watamura generated all these mutants before moving to UK when he was a research scientist in RIKEN CBS. He deserves the major credit for this achievement. I will later show you that use of these models led to identification of at least 2 distinct mechanistic pathways for tau pathology. This shows tau hyperphosphorylation in the triple mutant MAPTKA, mice carrying the S305N or P301 mutation. While the former shows only a little tau pathology, the latter shows intensive pathology already at 6 months, and the latter shows aging-associated pathology. This indicates that the triple mutant carrying P301S mutation exhibits early aggressive phosphorylated tau accumulation, but not the one with S305N. Also, use of antibodies which recognize various states of tau indicate that phosphorylated, oligomeric, and fibrillar tau accumulates in the triple mutant carrying P301S at 6 months, but not the one with S305N. This in vitro seeding assay also indicates that the triple mutant carrying P301S exhibits seeding activity comparable to 12 months old PS19, but not the one with S305N. These figures show astrogliosis, microgliosis, and synapse loss in the 2 lines of triple mutants. The MAPT mutation-induced tau pathology accompanies significant astrogliosis at 6 months, but very little microgliosis. Thus, microgliosis observed in the mutant tau. Overexpressing TG mice may be an overexpression artifact. Even at 12 months, these triple mutants do not exhibit significant microgliosis. Here is a quantified data. However, single-nuclear RNA-seq study provides more detailed information, as you will see later. To summarize these observations, whereas a beta pathology accompanies astrogliosis and microgliosis, TAU pathology induces astrogliosis, but little microgliosis. I suggest you be careful when you analyze neuroinflammation when you analyze your tauopathy models. This slide illustrates behavioral analyses of triple mutant MAPT knock-in mice using IntelliKage at 9 to 12 months. This system allows simultaneous observation of more than 30 mice, and the mutant mice showed FTD-like behavioral abnormalities such as elevated anxiety, repetitive behavior, impaired learning and behavioral flexibility, impaired sustained attention, and apathy-like behavior. We also observed regional atrophy in the triple mutant mice by MRI at hippocampus, hypothalamus, and amygdala at 12 months. Interestingly, both the triple mutant lines carrying P301S and S305N mutations exhibited brain atrophy in a similar manner, indicating a discrepancy with the pathological observations in these mouse lines at 6 months, as you saw in the previous slides. This discrepancy remains to be an open question and may imply presence of distinct pathogenic mechanisms. This slide illustrates the effect of a beta deposition on tau pathology of the triple mutant MAPT knock-in mice. Please note that a beta deposition exerts a greater effect on the cortex and hippocampus than hypothalamus and amygdala. In summary, the triple mutant MAPT line carrying P301S mutation reconstitutes the following phenotypes within 12 months. Tau pathology, phosphorylated, oligomeric, fibrillar. Synaptic loss, astrogliosis but little microgliosis. FTD like behavioral abnormality. Brain atrophy in hippocampus, hypothalamus, and amygdala. A beta deposition induced tau pathology, particularly in cortex and hippocampus. This is indeed a user-friendly model of FTDP17. Thus far, you have seen the phenotypes of the triple mutants, but now I would like to draw your attention to the double mutant line that carries S305N or P301S and intron 10 mutations. These mice allow analyzing the effect of aging and show the presence of distinct pathways that account for tau pathology. This slide illustrates that the 4-repeat tau is the most abundant in the S305N line, and then in the P301S line, where it is least abundant in the wild-type line. Consistently, the 3-repeat tau is the most abundant in the wild-type line, and then in the P301S line, where it is least abundant in the S305N line. Please note that 4-repeat tau is more pathogenic than 3-repeat tau in general. Interestingly, the number, width, and length of neuronal microtubules are significantly smaller only in the S305N line, indicating involvement of unique mechanism S in this case. Also, hyperphosphorylation of tau at 15 months is significantly higher in the S305N line than the P301S and wild-type lines. The 9-months-old PS19 line is a positive control, and the MAPT knockout line is a negative control. This slide illustrates phosphorylation, oligomerization, and abnormal conformation of these mutant lines at 15 and 30 months. Whereas the S305N line shows hyperphosphorylation without oligomerization and fibrillization, at both the time points, the P301S line shows pathological tau deposition only at 30 months. So the extreme aging can actually cause a beta-independent tau pathology in the latter case. Again, the S305N line exhibits hyperphosphorylated tau from 6 months through out to 30 months. In contrast, the P301S line shows hyperphosphorylated tau only at 30 months. This slide illustrates the effect of a beta deposition on tau phosphorylation in the S305N line, and it is notably high in the P301S line at 24 months, but not so much at 15 months. Importantly, this P301S line exhibits tau pathology only in the presence of a beta deposition at 24 months, and the beta-dependent tau pathology arises in an aging-dependent manner, implying the involvement of multiple molecular and cellular phases. And silver staining and in vitro seeding assay indicate the presence of fibrillar tau. Here is a single-nuclear RNA sequencing study showing that some microglial signatures are upregulated in the NLG FP301S line as compared with the NLG-F line. They include classical DAM genes, cytokine and chemokine response genes, and interferon responsive genes. This tau pathology of NLG-FP301S line disappears when they are crossbred with mice deficient in gene X, indicating that gene X product may be one of the mediators of this pathological transition. However, I cannot tell you its identity now, not because I want to keep it secret, but because the sample size is only 1, and there is no statistical significance for this observation. Please give us another 6 months to produce more reliable data. In contrast, the gene X deficiency exerts no effect on a beta pathology in this mouse line. Taken together, these findings indicate the presence of 2 distinct pathways that lead to tau pathology. One is phosphorylation pathway, represented by the S305N mutation carrier, and the other is the seeding pathway represented by the P301S mutation carrier. So, the first part of present study mainly analyzed the 2 lines of triple mutants, and it is thus necessary to investigate their pathological chronology and associated mechanisms in more details. In S305N double mutants, tau pathology was detectable at 6 months, after which the distribution remained consistent through 30 months. A beta-induced slow progression of tau pathology. P301S double mutant line exhibited no detectable pathology until 24 months. By 30 months, sparse neuronal tau staining occurred in cortical areas and hippocampus, but was strongest in thalamus and midbrain. a beta-induced fast progression of tau pathology. Therefore, use of the double mutants allows analysis of aging factors, but use of triple mutants saves time and resources. All the models are available from RIKEN Bio Resource Center. Several important questions remain to be addressed in the near future. Here are two of them. First, why do the S305 N-carrying triple mutants exhibit brain atrophy comparable to that of P301 S-carrying ones, where the former lacks any major pathology at 6 months? The former may exhibit accelerated pathology between 6 and 12 months, or another mechanism independent of phosphorylation and seeding activities may exist. Second, what are the cellular and molecular phases mechanistically involved in the A-beta-tau transition? Microscopic omics followed by reverse genetic approaches would help. Here is a list of major collaborators and contributors. Among all of them, these three young stars, Martha, Takahiro, and Naoto, made a major contribution. They have been extremely productive as well as reproductive. I also would like to express my deepest gratitude to the late Professor Yasuo Ihara, my lifelong mentor. Thank you for your kind attention. If you are interested in more details of AD research, may I cordially invite you to a book, Alzheimer's Disease Research Guide, that I published from Elsevier. I would be happy to share my PowerPoint presentation. Please feel free to contact me. Finally, here is an important message to the media.